preclinical phases, and in the discovery phase, it's through computational modelling, and this is the QSAR models, quantitative structure activity relationship. And basically, what this is a giant database of drugs that have been used, and all the clinical and testing, everything that's been done in the literature is put into this database. So if these chemical libraries have been tested for safety, that information goes in. If these chemical libraries, the drugs have been tested for efficacy against diseases, that information goes in. So, so it's a generation of these mathematical models, and then what you can do is input new structures into these models and be able to get an idea of their physical, chemical, biological and environmental fate or properties of these new compounds. So it's just another way of screening. So if you know that in a particular chemical structure that a particular side group is toxic, then you can say, well, actually that compound will be likely to be toxic, and remove it from further testing. So that's in the discovery phase. In the preclinical phase, they use more complex and physiologically relevant models. There has been a huge push to move things into in vitro testing using cell-based models, in vivo testing using animal models, but even before using mice to look at these, to do safety studies, there are other in vivo animal models such as insect models and zebrafish models. Have you come across these? So there are other sites of, and I'll talk about some of these models today. And then there is the PK/PD study, so the ADMETs, the absorption, distribution, metabolism, elimination. And the reason why that is important is because if you've got a drug and it's in the body, it is removed really quickly or degraded really quickly, then there is almost no point screening it in in a mouse model, for example, because you're setting yourself up for a fail. So really what you want to make sure is that you know how long the drug is going to remain in the body and for how long. Because if you've got a drug, say for example, where initially there's a high concentration for the first 6 hours and then it degrades, then you could be thinking, okay, in order to maintain that drug at the level that I want it, then I need to dose every 6 hours. So that's why the PK studies are really important. It informs you, one, how to deliver the drug, which route is most efficient, whether it's oral or by injection, and then how long it stays and where it goes in the body. And then in the clinical stages, there's the human safety trials, which I'm not going to focus on today. So virtual screening. So I'm going to basically go through this diagram here. So like I said, QSAR uses laboratory collections and literature data on all chemicals that are used, that have been trialed. So they collect all that data. So like I said, it'll be any safety data that people might have done, any safety screening, any efficacy screening, and any other information if there's any PK studies done on the drugs. They all go into this QSAR modelling. And so once you've got this model, what you can do is you can input like millions, literally 10 million chemical structures into this model. So these large chemical libraries into this model. So you're screening virtually, basically. And then it goes into the model filters it out, and you get a lot less chemical structures, something between the region of 10 to 1,000, which is much more manageable than 10 million. But this approach actually allows you to screen a much larger chemical library. Can you imagine making 10 million chemical structures? I know my chemist colleagues, some of them are, so they're like, oh, some things can take days, other things can take months, other chemicals you just can't make. So you wouldn't want to really try making that many chemicals, but if you can do it in this virtual screening method, then you can down select to a manageable number. These manageable numbers can be synthesized, and then the first thing you would do is in vitro assays. You would do in vitro assays which would look at cytotoxicity. Hi. Morning. That's all right. Come and sit down. In vitro assays, so you would look at safety, or you would look at and you would look at efficacy. So looking at efficacy, testing the drug against whatever disease it is that you'll want to identify inhibitors for. And if it passes in vitro assays, then you would consider doing in vivo assays. So this is how the QSAR model works. The advantage of QSAR is that it's low cost. You potentially get a higher hit rate than traditional high throughput screening because obviously you're starting with a much larger chemical library. Okay, I think I can use this. So I just wanted to give you a couple of examples where these QSAR models have been used, and one is for the infectious disease malaria. I'm not going to go into malaria. I am pretty confident you all know probably more than me about malaria. But in this group here by Zhang et al., they started off with a data set of 3,133 compounds, and these compounds were reported as either active or inactive against Plasmodium falciparum. And so they built this QSAR model using chemoinformatics. So basically, it's just like it's like a DNA. It's like genomics, chemistry, and information about chemical drugs. And they use these. They use programs such as Dragon descriptors, amongst others. And then they applied the virtual screening of the ChemBridge database. So they based their model on these 3,133 compounds because they were tried against Plasmodium falciparum. But then they used all the chemical structures that was in the ChemBase database. So like I showed you in this, so they modeled it here, the 3,133 compounds here in the modeling, and then they applied the ChemBridge database to the model here, and they did the virtual screen. So I'm just talking about, sorry, what was your name? Yeah, Faisal. I'm just talking about safety screening and in the early discovery phase, which is through modeling. So that's what this is, just to set the scene. So from that, they identified 176 antimalarial compounds, which they went ahead and screened, along with 42 that were potentially inactive. So that 42 was like a control set. And then out of this, they found 25 compounds that had antimalarial activity, which inhibited the growth of Plasmodium falciparum and showed very little or no cytotoxicity in mammalian cells. So that was obviously a bonus. And the modeling correctly predicted the 42 inactive compounds, which were also validated experimentally. So what these studies did was result in new chemical scaffolds as a promising starting point for the development of antimalarial agents. I think this is an amazing way to use the information that is already out there. If you think about the research that everyone does, there's so much information that is out there. To actually extract all that information and use them in databases like this to inform future drug discovery programs is fantastic, especially using the different aspects. So using safety testing, efficacy, and also and others to inform the model. So the second example that I wanted to give you was on tuberculosis. Again, I'm sure you guys all know about Mycobacterium tuberculosis. So here, this group here, Gomes et al., they used QSAR models to design a new series of chalcones derivatives. And so details of all chalcone compounds which showed activity in vitro against Mycobacterium tuberculosis was obtained from the literature. They did a rigorous data curation followed by structure-activity relationships. And then based on these structure-activity relationships, they were able to employ and design new chalcone derivatives with optimized anti-TB activity. So they were able to do this, and in parallel with the QSAR model, they were able to identify further new chalcones. And from these, 4 were found to be really potent as the drugs that are used to treat tuberculosis at this point in time, and they demonstrated low cytotoxicity in mammalian cells. So in the early drug discovery phase, it's quite often researchers don't think about drug safety because they're thinking about how to make a chemical that is going to best target whatever protein or whatever it is in the cell that they want to. So they want to make the optimal chemical structure, which is the best starting point if you think about it, because then it will bind and it'll be inhibitory. Well, that's what you're hoping. So not necessarily, safety doesn't necessarily come into it until a bit later, and then really what you find is that the chemical structures are made, and if they're not safe, then you have to go back and start again. So using this sort of modelling can really help move forward a bit quicker in the process. So now I'm going to move on to talk about in vitro safety screening of drugs. So in vitro safety screening, it generally uses cell lines to detect cytotoxicity of compounds, and they're fantastic because they're cheap, they're reliable, depending on the method, and they're very easy to do. And you're going to tell me if they are or not after the lab. I'll ask you tomorrow afternoon if they're easy to do. This reliable, depending on method, actually is quite interesting because in my lab we do cytotoxicity screening. My collaborators, chemistry collaborators, give us a lot of different inhibitors, and some, as soon as we put them into our cell culture models, you can see the cells dying. So we do quite a bit of cytotoxicity screening ourselves, and we do find that they are quite variable, so it really depends on which method you're using. They are high throughput because you can do monolayers in 24 well plates, and that's what you're going to be doing this afternoon, or you can do monolayers in 96 well plates as well. So if it's in 96 well plates, then you can see how high throughput it is. You can probably screen 30 compounds because you need to do things in triplicate in each plate, and you can do a number of these different plates. And so it's high throughput, dependent on sensitivity and scalability. And the most important thing is that it reduces the need to screen using animals. If you think about it, if you had 2,000 compounds, then you would be using thousands of mice to screen in animals. Whereas that you can do the first preliminary screening in cells, and then use a much more reduced number of animals and experiments. So basically, in vitro cytotoxicity looks at cell viability. And so these are, over here, are the different methods that can be used. And they're all based on enzymes that are released when the cells are dying. So membrane permeability, enzymes are released into the supernatant. Or they're based on looking at some components of metabolism. So I've got some methods here: tetrazolium reduction, resazurin reduction, protease markers. These measure aspects of general metabolism or enzyme activity as markers of viable cells. So if a cell is viable or alive, then obviously its metabolism is ongoing. If a cell's dead, then it can't metabolize things. That's essentially the principles. And then there's ATP detection, so measurement of ATP using firefly luciferase. And then you have other methods such as flow cytometry, high content imaging. Now these are much more expensive because you need much more expensive kit to look at the data, and it's not as high throughput. So traditionally, we normally use these methods for cytotoxicity testing. I'm just going to run through some methods and let you know which ones you're going to be having a look at. So the first one was the tetrazolium reduction assay. And the tetrazolium compounds, a number of them are used to detect viable cells. And MTT is one of the most common and one of the first assays that was developed. And it was developed for a 96 well plate format. MTT, this is the chemical name for MTT, dimethylthiazole diphenyl tetrazolium bromide. It's much easier to say MTT, we'll stick with that. And so you add the MTT substrate to cells, and basically what happens is MTT is yellow, as shown here. The mitochondrial dehydrogenase, if the cell is still metabolically active, converts MTT to purple formazan. And the MTT isn't fluorescent, but the purple formazan, you can measure the formation of formazan using a plate reader. And so this is the reaction, and this is the output. So you can see here, there is hardly any purple formazan here. So these indicate that the cell is not metabolically active because you're not getting conversion of the MTT to purple formazan. Whereas here you can see lots of purple, indicating that you've seen a lot of cell death here. So resozurin reduction works similar kind of principles. This is one test that you're going to be doing this afternoon. Well, you won't be doing this afternoon. You're going to set up this afternoon, ready to do tomorrow. So resozurin has been used for decades. It's quite an old method, and it's usually a reliable method, and it's cheap. So resozurin, when it dissolves in buffers, it's blue in colour and non-fluorescent. So metabolically active cells reduce resozurin to pink. So here is the reaction. Here's resozurin. Viable cells use NADH to produce resorufin. And resorufin is pink with high fluorescence. So you can see here, lots of pink here. These cells are quite happy, whereas you see blue here indicating that, that the cells aren't quite so happy. So resozurin is slightly more sensitive than the MTT assays, and it can be used for high throughput. One of the things that I will say is that a lot of these reagents are sold as kits from companies, and the companies have tweaked some of the enzymes, some of the catalysts that are used in the reaction so that they perform a little bit better. So you get a lot of different variations of this. So I've talked about metabolism. Here, the protease viability marker is a measurement of protease activity, so measurement of enzymes that are present. So cells use a permeable fluorogenic substrate such as GFAFC. Here's a longer name. And this penetrates the cell. It selectively detects protease activity that's only present in viable cells, because if the cell's not viable, then the proteases kind of disappear. Cytoplasmic aminopeptidase activity within the cell removes the glycine and the phenyl amino acids from the GFAFC to produce aminofluorocoumarin, which is here. And this generates a fluorescent signal proportional to the number of viable cells. So they're all very, very similar, these assays, in how they work. The ATP assays, I have to go through this one because this is probably the most common, because all what you're detecting is light. So essentially, it uses 5-luciferase, and it is incredibly high throughput and very quick. So ATP is widely accepted as a valid marker for cell viability, and the loss of the membrane results in the cells losing their ability to make ATP, and any endogenous ATPases rapidly deplete ATP from the cytoplasm. So ATP detection reagents contain detergent. The detergent lyses the cell. ATPase inhibitors are present to stabilize the ATP that's released from the cells, and then it contains luciferin, which is used as a substrate, and luciferase to catalyze the reaction, releasing light. So you can see they're all quite simple, but very effective. So this is a test that you're going to be doing as well in the practical. This is crystal violet. So crystal violet is a dye that binds to protein and DNA. Now what this test is actually relying on is the fact that when in a monolayer, dead cells will actually float and lift off because they're not adherent, they can't stick to the plastic. So this assay is looking at the number of cells that are adhered and making the assumption that the adhered cells are still viable. So this assay probably is, well, I was going to say it's not used as much as some of the other ones that I talked about, but actually in my lab, I don't know if you know Nicole, my postdoc, she's been using crystal violet recently and has been telling me how good it's been. So there you go. It just depends on what it is you're looking for. So crystal violet will bind to the protein in the DNA. So adherent cells detach from cell culture plates during cell death. So crystal violet can be used as an indirect quantification of cell death and proliferation. So here you can see that here's two drugs. This is from this paper by Yan et al., where they looked at distant cytotoxicity screening of remdesivir and hydroxychloroquine. And you can see that they high to low drug concentrations. And you can see that remdesivir at high concentrations, there is hardly any cells, because if the cells are in the wells, then you see this deep purple color. Hydroxychloroquine, you can see even at the highest, they only went up to 100 here, whereas they went up to 170 of remdesivir. You can see that there is no cytotoxicity. Now, the final assay that I'm going to talk about is lactate dehydrogenase, and this is, you're going to be doing this assay as well. So you're going to test 3 of these assays, and this is the third one. So LDH measures the level of plasma membrane damage in a cell population. So LDH is a stable enzyme that's released into the cell culture as soon as there is membrane damage. And actually LDH is the assay that we routinely use in my lab to look for cytotoxicity. And we actually buy it from Sigma. So you guys are going to be using the same kit we use upstairs. And there's 2 reactions. First of all, the LDH reduces NADH to NAD, so NAD to NADH by oxidation of lactate to pyruvate. So that's the first step. And then the second step is this NADH is used in this reaction, and you'll recognize this reaction because this is the tetrazolium salt catalyzed to the formazan salt, and so the red is what it is that you measure. So this is just coupling an enzyme to this reaction for the resirufin or the MTT assay. But because it's detecting LDH, this is actually a lot more sensitive than the other ones, and a lot more accurate. So that's the LDH assay. So these are just some of the in vitro toxicity assays that we can use, and it really reduces the number of compounds that we will use to go on to the next safety screening. So the next safety screening is in vivo. So the advantages of in vitro toxicity testing, but there are limitations, because you're looking at a monolayer of cells. I mean, we're not made up of a monolayer of cells, are we? We're made up of complex organs, different types of organs, different types of cells. So therefore, it doesn't really reflect the toxic effects in whole body systems. Chronic toxic effects can be very difficult to observe. So when you're doing these toxicity testing in the monolayers, you're looking at an effect straight away. But what about longer term? What about longer term exposure to drugs? So these chronic effects are more difficult to observe, and we can use it as an indication of getting rid of really super toxic drugs initially, but it can't be used as the final basis for toxicity assessment and risk assessment. So before we go into mice, there are alternatives that you can use that are gaining a lot of momentum nowadays. And these are, I'm going to talk about 2 in particular: the zebrafish, Dani or Rio. Have you come across zebrafish? They're pretty cool. And Galeria melanella. Have you come across Galeria melanella, wax moth larvae? No? Oh, you're going to hear a lot about that this morning. Anyway, so this is a zebrafish, and this is obviously Galeria melanella. So we'll talk a little bit about these models. So the zebrafish model, the reason why these models have become so popular is because it's low in cost, low in maintenance. They can be very quick, they can be high throughput, and they can be accurate. The other reason is there are very few ethical issues. Ethics is really important, and I'll talk a bit more about this in my lecture with when I talk about mass experiments. But my last ethics application was 150 pages, going strong. You have to go into that level of detail about what you're going to do. So in order to use in vivo models where ethics don't come into play is fantastic if you can house them. So the zebrafish model actually is, we do have that at UWA at the Biomedical Research Facility in Shenton Park. So they do have the zebrafish model if you want to use it. So anatomically, genetically similar to humans, there's a large toolbox. So I showed you this image here. You can see straight away that this zebrafish is glowing. And so there's a lot of markers. There's also a lot of, like in mice, you can get a library of mutant mice that have mutations in different genes. Well, same thing for zebrafish as well. So it has a large genetic toolbox. Optical transparency of the embryos and larvae, enabling large phenotype-based screens. So here you can see, I've just highlighted some of the things that you can see. So here is a nice normal zebrafish embryo. Here you can see that's a dead embryo. Here you can even see the egg cell fragments. So this is obviously a 96-cell plate with individual zebrafish eggs in there. Here is an unhatched embryo, and there you can see this embryo is just hatching. So it's quite visual. You can actually see what's going on, which can be very powerful. So the occurrence of many pathological processes that resembles humans. However, the disadvantage is that it has only a moderate level of predictive and translational value. We only are starting to discover this now because drug screening is coming through the model, and then we can see where the ins and outs are of the model. So it's only moderate. But it has been used. In TB, they've used it to look at some drugs, this Audace et al. So this is some of the results that they got. So what they did was 2 experiments. First experiment that they did was to look at efficacy. So here they injected drugs robotically, and then they treated, they didn't inject drugs. They injected the pathogen, Mycobacterium marinum. So Mycobacterium marinum is a natural pathogen of fish. So that's what they use to infect zebrafish, because of course Mycobacterium tuberculosis won't infect zebrafish. This is one important thing to consider about animal models, is the animal appropriate, and is the actual model, the disease model, appropriate to humans. So that's a really important consideration. So in this one, they used Mycobacterium marinum, they infected zebrafish, and then they treated with drugs. And look, so it's GSK, GlaxoSmithKline, 10, 14, 43, and 19. You can see this is a relative inhibition. And you can see that drug here looks like it's effective. Here, not so much there, but this one, there is no inhibition here at all. And so, as well as looking at efficacy, where they showed these 3 drugs were efficacious, they also looked at uptake. So remember what I was saying about the drug, how long it lasts in the system is really important as well. So they did this for 6 days. So basically they did drug treatment, and then they sampled the yolk, and then they did mass spec to see if the drug was in the system after 6 days. And look here, you can see that GSK10, you saw a large amount of drugs that are in the system, and these are less so, but they're still in the system. But look, this is probably why this isn't very effective, because there is no drug in the system. So that's why the PK studies are really important, because you can get an idea of what is happening. So the Galleria melanella, wax moth larvae, this is considered also to be a non-animal technology and accepted as an ethical alternative for research. The Galleria melanella models since 2010, if you look at the literature, the amount of people screening in Galleria has gone up exponentially. And it's because they're actually quite easy to maintain. You don't even really need a true insectary. You just need like a 37-degree incubator. And they reflect some mammalian complexities. They possess an innate immune system, but not an adaptive innate immune system. This innate immune system is structurally functionally active to similar to mammals. In the UK and other countries, Galeria melanella larvae aren't regulated, so again, you don't need ethics, reducing the cost of studies, enabling the system to be used flexibly. And they used this 6th instar stage here. And this, in this stage, they don't feed either, so you don't have to give them food either. So they're really cheap, cost-effective, and so they've been used as an alternative model to look at pathogenicity of pathogens and also as efficacy of drugs. So here is one study that was done by Coates where they looked at orchidaic acid, which is a toxin produced by certain dinoflagellate species such as diuretic starfish poisoning. And so what they did was they wanted to see if screening the cytotoxicity of orchidaic acid in Galeria melanella was equivalent to looking at it in mice. And so they screened different doses. You can see here the different concentrations, and you can see that there is a dose-dependent effect. And the conclusions were that using the Galeria model, it was comparable to LD50. LD50s are lethal doses, so 50% of lethal dose. In rodent models, they display broad symptoms of immune cytotoxicity and oxidative damage, and you can see that's here. So here, this is moribund Galeria, but you can see that these Galeria, that they are undergoing some stress, and the drugs are having an effect. And so it looks like you could use Galeria melanella to look at cytotoxicity. But ultimately, you still have to go and have a look in rodents. Rodents are the ones that are used the most for safety screening, and also looking at PK. Now, mice are used, but actually my collaborators tell me that rats are more of a physiologically relevant model to humans. So really, when it comes to safety and PK profiles, usually the rat model is used. And the biosafety assessment, certain parameters required, the PK profile, they look at the immunogenic response. You don't want to have induce a huge immunogenic response to the drug because that could hurt you as well. And you can do biochemical and histopathological analysis. And then for the evaluation of efficacy, you need an appropriate model. You need to think about the routes of delivery of the drug, and you need to think about routes of infection challenge, and all these three will determine the output. So not only do we screen in animals for drug safety, but companies are offering other safety screening. So there are certain human targets that drugs that have been identified, where if the drugs have off-target effects of these human drugs, then you can get cytotoxicity. So what these companies have done is worked with the big four major pharmaceutical companies, and they selected 44 human targets. And they essentially have the targets in a chip kind of assay, and they will run the drugs through to see if the drugs will bind to some of these human targets. And so they call it Safety Screen 44, and this is the panel that was done by Eurofins Pharma Discovery. And so you essentially send your drug to them, they do the screening, and they say, well, actually, that binds to one of these, and that has really bad kidney effects. Or this binds to that has really bad liver effects. So you can see that even before you go into animals, there's a lot of tools that are available to look at drug safety. So PK, I'm not going to spend too much time on this because you've done most of this already, but it's really important to understand the kinetics of absorption, distribution, metabolism, and excretion. If we know this, then we can design an experiment to look at drug screening in animals under optimal conditions, giving the drug the best chance to show an effect. There's been so many animal experiments that's been done in the past where it's not been optimally designed, so we've probably missed certain drugs because they didn't have an effect in mice, but they didn't know if the drug was still active in the mice. They just tried it. So to avoid that, that's why the PK and the ADME studies in particular are really important. So ADME, so absorption, so distribution, metabolism, elimination. Again, I'm not going to really dwell on this now because you've done this. So, but we do need to know this information to design a good animal experiment. So just in summary, without safety guidelines and safety studies, the use of drugs in the general population is potentially lethal. And so for the protection of the general public, obviously FDA is one, but Australia has the TGA uses similar kind of guidelines, same as the UK, similar kind of guidelines to protect the general public and make sure that drugs are tested for safety. So the drug discovery process includes several safety screening points from early drug discovery stage, so the QSAR modelling, in the preclinical stage, quite a lot of drug screening, in vivo drug screening happens, and then also in the early stages of clinical. QSAR virtual screening are a really good way to high throughput screens for discovery of drugs that may be more safe. But obviously this relies on large databases of information, which we are getting. So, you know, getting better at this, but it's still in the early days. In vitro screening is really important, screening in cells, because they're an excellent indicator of cytotoxicity. Alternative models, always consider the alternative models when you're looking for drug safety screening and before going into rodent models. So that's my first lecture. Oh, I put this up because this is why we start off with tens of thousands of compounds at the top, and after you've done the early drug discovery and you've done the early efficacy, you lose a lot, and then even at the preclinical stage you lose a lot, and that's why at the end of all of this you end up with very few drugs that are actually really good. But all these steps are there to protect the general population. But that's the end of my first lecture. I don't know how you guys do it, whether you carry on or whether you want a little 5 minutes. I don't mind. I'm going to have a drink first though. So I'm going to have some water. Have you got any questions? Yes?
So what's the ethical line with animals? Because I would consider a fish an animal.
So yeah, that's a really good question actually. With the zebrafish, it's up to the stage where they start feeding. So when they start feeding, then ethics comes into play, which is why a lot of studies are done in the early embryo stage. But with the Galeria, again, they're not feeding, and you don't want them to carry on to the moth stage anyway, because you know, if you're giving an infectious disease to a Galeria that turns into a moth, you're asking for all sorts of trouble, because literally they're. I haven't got any pictures. I should have. I've got. Collaborators. Nicole went over to their labs at Macquarie University and to have a look at some of our bacterial mutants that we've got and also to screen the drugs that we're looking at. And so she went over there, and they basically grow them up in like jam jars essentially, and they have like loads of these jam jars with them in the different stages, and then when they get to the 6th install, that's when they use it. And when they use them, and literally when you have samples of 10 larvae, and I'll show you how they inject it in the next slide anyway, but once they've injected it, they just put them in a Petri dish. So can you imagine if they turned into moths in a Petri dish infected with a dangerous pathogen that isn't, so when it comes to like some of these models, you have to think about what containment that these models are going to be housed in. So Burkholderia pseudomallii, I might as well give you the lecture. I don't know if you know much about it actually. No? You probably, well no, I don't lecture, so I lecture in the MID, so you don't, I do a 3rd year where I talk about Burkholderia pseudomallii, but it causes melioidosis. It's endemic in Northern Territory and in Southeast Asia. It resides in the soil, and it's, because it resides in the soil in Australia, it's classed as high containment PC2, so I can handle it. So we handle it up in the Marshall Centre in the lab up there. But in other countries, it's actually a PC3. So before I came here, I worked at Defence Science Technology Laboratories in the UK. So preclinical studies, in vivo testing. So recently, I won't say recently, I'd say in the past 30 odd years, in vivo testing has been, and using animals in experiments, there's been a real clamp down on it, and the whole ethics behind it is a lot. And so you have to have a very good reason why you want to work with animals, why you're going into it. You have to have an appropriate model. You have to ensure that your numbers are correct, and you have to make sure that you're making the most out of all experiments. So if back in the, like, animal activists obviously are against it, but how many of those animal activists do you see volunteering to take experimental drugs? You know? so it's like, well, because that is ultimately what we would need to, we have to make sure that it's safe for people before the drugs go out there. I mean, you know the consequences of what happens if you don't. So there is a huge regulatory body around animal experimentation, and wherever possible, we try to minimize it, which is why these alternative models have come about. And so I'm going to talk quite a bit about the alternative, that's why I'm talking quite a bit about the alternative models, because they've been increasingly used. And then I'm going to talk particularly about the mouse model, and talk you through a case study of my own group and how we've developed the mouse model to screen drugs against Burkholderia here. So you should understand from this lecture, you should understand why animal models are important, to list the advantages of alternate animal models. I'm going to talk again about the Galleria melanella and the zebrafish models, but this time why they're good models for infection and screening on novel drugs, list the requirements when selecting an animal model. And then I'm going to talk to you about the mice infection models for Burkholderia pseudomallei. I'm going to talk about two particular models, one that causes chronic and one that causes acute melioidosis, and then list the 3 R's and why they're important. So the importance of animal models, well, they are really important for us to understand diseases, what the bacteria or whatever pathogen, what effect it has on the body, how the body responds to the disease, and this helps us to expand our knowledge on diseases such as cancer and infectious diseases. So this is a really important point, is that, you know, for us to understand a disease, we have to be able to look at it in the model to see what reactions, and then using these models to test drugs and other therapeutics, and of course testing of diagnostics as well. So here are the regulatory considerations. So using animal models is regulated at the national, regional and local regulations and policies. So national have a framework of all the things that you need to do when working with animals. Here in Australia, I find it quite interesting because in the UK there's a home office regulations here. Each state has different regulations, and when you do the ethics course, it's quite interesting how each state considers different animals as whether they're animals or not. So like in Victoria, fish are considered animals, so you need ethics to work on fish. In WA, fish aren't considered animals. So it's quite interesting, and that depends on who is on that committee and how they recognise them. So there are two big considerations. There is the animal welfare, and that is what the committee is most focused on. So the Animal Ethics Committee here at UWA, they're composed of a chair who is a scientist. It's composed of a scientist that is in, say for example, that is in biomedical research, and then it's composed of another scientist that isn't in biomedical research. They have a lay person who they bring in, and that person doesn't have any idea of animal experiments, and then they've got another person that's from a vet as well. So they have this committee, and they will assess each individual ethics application. And so the biggest concern is animal welfare, and that you're managing the pain that the animals are going to go through, or that you're managing, if you're giving them an infectious disease, that you're managing that disease and that you have the appropriate humane endpoints. So that's why the 3 R's are really important. This is replacement, reduction and refinement. So wherever you can, you need to replace the use of animals, so hence why this big drive of these other alternate models, hence why cell models are used quite a lot as well. Reduction, so using the minimum amount of animals for your study. However, small isn't necessarily always good, because you have to, from your experiment, you want to get statistically robust data. So you need to ensure that the number of animals that you're using is going to give you that statistically robust data. So you want to reduce it, but you want to make sure that you make the most of the experiment and get the data that you need, and that is statistically significant. And then refinement. So refinement's basically how you can improve the life of the animals. So if there is one particular technique that's a bit rough on the animals, then what can you find an alternative way of doing it? And then there's scientific considerations. There is research objectives, so response to infection, host susceptibility, size, cost, anatomy, safety, genetics, and the availability of reagents. So again, like that zebrafish model, you use Mycobacterium marinum because you can't use Mycobacterium tuberculosis because it won't infect zebrafish. But does marinum and zebrafish equate to humans and TB? See, that is the ultimate question, is that does the animal model reflect what you see in humans? So that is really important. So therefore, the animal species and infectious agents is really important. But first, before I go into our mouse model, I'm going to talk about alternative models. So you can see, this is just some of them. It's not all of them. The Drosophila, the fruit fly, has been used. They obviously talked about zebrafish. C. elegans, Rhabditis elegans, has been used quite a bit. Talked about Galeria. The two-spotted cricket has been used, and the silkworm. And I've got this one here, the Madagascar hissing cockroach. That's my favourite. You're not going to believe it. These cockroaches are like half a hand length. If you go on the internet and get a picture, it's like they're this big. And like I was shocked to see 2 groups, I know them both, in America, actually use the Madagascar hissing cockroach to look at Burkholderia pseudomallei virulence. And I was like, how are you doing this in PC3? Because they would have had to do this in PC3. And more importantly, is how are you handling it without going, ugh? But yeah, so that they have been used as well. And the reasons for using these models is cheap, simple to use, and can offer high throughput screening. You can use lots of them. There's no ethics, or ethics are minimal. And so these models have been used to investigate molecular mechanisms of various human pathogenic bacteria, and also screening novel drugs. So that's it. That's mainly the reason. Lack of ethics, not limited by ethical regulations, cheap, simple, and high throughput. Although I don't see how this can be that high throughput, really. But some of the others are. These ones here you can use in 96 well plate formats. So I'm going to talk about Galeria melanella. So like I mentioned earlier, the use of Galeria melanella has shot up in the past like 10, 15 years. And the screening of drugs shows similar results to that in mouse models. And I'm going to show you some data from my own lab using Galeria melanella as well. So they have a number of attractive features: easy to use, no ethics approval, low in cost, because like I said, you just need an incubator that will house the larvae. And the other advantage is that it has the innate cellular immune cells, which are similar to neutrophils and macrophages. It does lack the adaptive immune response, so there are limitations. And it produces antimicrobial peptides such as opsonins and PRs, which, so again, similar to humans, but not quite the same. So that's why you're never really going to pull away from mouse or rodent models or an appropriate animal model, because these are similar but not exactly the same. It possesses the hemocytes, and these are what are similar to mammalian phagocytes. And it's been used as an infection model for a number of different bacteria. So how is this done? Well, here is the larvae, and usually you inject straight into the hemocoel, which is the main body cavity of the Galeria, directly into its proleg here, like so. And sometimes, in order to make it really clear, you move the larvae underneath the tip so that you can inject really accurately, like shown here where the injection site is. That is the proleg here. And then the melanization is what gives an indication of its health. So you can see this is moribund, so dead, whereas these are not very well. So you can do dead live, or you can do a scoring system of a little bit of melanization or a lot of melanization. So there's different ways for you to measure the output. And in some experiments, some people also weigh as well. And so some colleagues of mine have used the Galeria melanella model to look at Coxiella burnetii. Do you know about Coxiella burnetii? Coxiella burnetii causes Q fever. And Q fever, Q stands for query fever. I don't know if did you guys get to see Steve Graves' seminar? He came and gave a seminar a little while back now, I think back in, it was in end of June. And he works on Q fever. And basically it's a pathogen that infects cattle, and human are usually accidental hosts, but it's caused quite a lot of huge outbreaks. It's quite prevalent here in Australia, especially in the rural communities where there's farms. In the Netherlands, something like 4,000 people, or an outbreak of more than, I think it's more than 4,000 or 40,000 people over a period of 10 years, and they're mass culling of cattle to try and get it under control. But it causes, most people won't even know they've got it, but in some people it causes chronic disease. Anyway, so some of my collaborators, so Izzy here, she works at DSTL, she was my PhD student. She actually looked at Coxiella burnetii in the Galeria model. And here you can see that the groups that were given PBS, that's a control, that's just PBS, buffer and heat killed, you can see they survive. But the ones that were given Coxiella burnetii, you can see all succumb to infection. So it's a model that you can use to look at virulence. So if you've got some mutants of Coxiella, you could use the Galeria to see if the mutations make it less virulent. It's also been used to test a number of different antibiotics. And I've put this table here for, if you're interested, you can go and have a look. But you can see that there's lots of different infections that have been looked at, and this is only just some of them. What they found was that the results that are coming out of Galeria is very similar to what you see in mice. So I'm going to tell you a bit about the work that we've been doing on Galeria. My postdoc, Alex, she used the Galeria melanella to screen 2 experimental compounds that we've been working on, SF235 and AN296. And these compounds target a virulence factor that we've identified that's present in a number of different bacteria. The main ones that we've been working on is Coxiella burnetii and Burkholderia pseudomallei, and it's called the macrophage infectivity potentiator, the MIP protein. So we screened these drugs. We did in vitro screening first. So we had a look at them in cell infection models, and we showed that the drugs actually reduce the survival of bacteria in cells. Now, the MIP is a virulence factor. So basically you take it away and makes the bacteria less able to cause disease, but it doesn't kill the bacteria. Okay, this will come important later on in the animal experiments and how we've designed the animal experiments. So really what we're hoping is that these compounds will be an alternative therapy to antibiotic resistance. So you could use the compounds on their own. It will make the bugs less able to cause disease, and your own immune system can kill it off. So that's one way that you could use it, or you could use it in combination with antibiotics. So then making the antibiotics more effective. So they're the two ways that you would use the inhibitors. So what we did was, and we did this in collaboration with a group in Monash University, because they won't let us have Galeria melonella here in WA. So Galeria melonella is considered a biohazard. WA are saying that they don't have it in the environment, which is not true. They do, because it's present in all beehives. And so, but it's not, we're not authorised to use it here. So we're actually about to undergo the painstaking process of fighting this to get it here, because it is a really good alternative model. So we have to request that we can use it and undergo a lot of painful paperwork to do that. But so we did these studies with our colleagues in Monash University, and what she did was she tested AN296 against Coxiella burnetii using Galeria melonella. And so what you can see here is the DMSO, which is essentially the vehicle that the compounds are dissolved in. You can see they succumb to infection, but with AN296 and with SF235, we see protection compared to the untreated. And in cells, what you can see here, this is the untreated. In the pink is the Coxiella. They grow in like vacuoles in the cell. Blue is the DNA. You can see that there's lots of pink, or it's meant to be red, but it comes out as pink because there's so much of it. There's lots of Coxiella, but with SF235, and particularly with AN296, you can see there's hardly any bacteria left. So this is, so these drugs look really quite nice. And we're actually moving into looking at these drugs in animal models against Burkholderia. So zebrafish as a screening for TB again. So, so I've talked through the advantages of the zebrafish. One thing that I didn't mention was its high fertility and rapid development, which means that it's really quick to get a lot. Even with Galeria, like you have to grow the larvae to that certain stage, so that can take a few weeks. Zebrafish, I think you're looking at days. So the advantages of Mycobacterium marinum, has a shorter replication time. You know that growing TB, I think, takes weeks. Whereas marinum, I think it's less than a week. But it's still not as quick as E. coli, but it's a bit quicker than TB. Lower safety regulations, and in zebrafish, the marinum causes TB-like disease, and that's what's really important. So here, in this picture here, you know, you get the tuberculosis, you get the necrotic lesions. Well, here you can see the lesions here on the zebrafish here and here. So that's what's really important, which is why the zebrafish gets used, marinum zebrafish model gets used, because it is reflective of TB disease in humans. So marinum actually can cause the skin lesions in humans as well, so it does infect humans too. So in this group here, they, I've not got the reference to this group, I missed it out. They use the models to differentiate between toxic and non-toxic compounds, and so they treated infected embryos by adding the drugs directly to the fish water. So first of all, they will have injected with marinum. Oh, sorry, no, this was just looking at drug toxicity. And so they tested drugs at a single concentration of 10 micromolar, and 3 days post-infection, bacterial loads in each embryo was quantified using fluorescence readout. So here is, in the orange here, is marinum, and this is these embryos have not had any compounds, so you can see lots of bacteria growing. Here, this is where they've injected an active compound. You can see that there is no bacteria growing, indicating that the compound is active. Here, the non-injected, you can see that there's no bacteria at all. Toxic lethal compounds, you see this circling of the They don't even come out of their eggs, really. So in this experiment, they trialed a number of different drugs here. And you can see that some treatment with these drugs here resulted in significant reduction of bacterial signal. So essentially, they just looked at the fluorescence of bacteria as a readout, and there was reduced in these drugs. But treatment with approved anti-TB drugs, such as streptomycin, kanamycin, and so on, at 10 macromolar didn't reduce the bacterial load in infected embryos. And so you can see that you need to use much higher concentration of these drugs than some of these new ones where they are showing an effect at lower concentrations. So they used this model to screen 240 compounds, and they identified 14 that were highly active. If you were to screen 240 compounds in mice, then you would be using 2,400 mice. So that is the cost and everything goes right up in with mice. So you kind of want to go into higher order animals with a much reduced amount of drugs to screen, just to keep the numbers down. So there are many things that you need to consider when you're selecting for animal models for research. First of all, is its appropriateness and comparable to humans. So doing things like PK studies in rats because it's physiologically more relevant to humans than mice, for example. The transferability of information, is it what you're going to get, is it applicable to humans? So again, if you've got a infectious disease model, is that model showing what happens in humans? Because if it isn't, then you're not going to be able to use that information. Genetic uniformity of organisms. So a lot of the animals that we use, they're inbred. Background knowledge of biological properties. Cost and availability is a really important one. If you think about, these are just some of the animal models that have been used. Mice, you can house them in groups of 5 in a cage, whereas marmosets, you have to have them in pairs because that's how they are happy. Because in the wild they live in pairs, a female and a male, and so then, and also with like the marmosets, their cages are really quite large. Other animals that have been used in melioidosis research, goats have been used. In fact, goats were the first. Melioidosis in Australia in the 1950s was discovered in goats, and the farmers were accidental hosts. And then there are hamsters. Hamsters are they are quite cute, and they have a lot of fur. But hamsters have been used for melioidosis because they're extremely susceptible. So again, you want to use an animal that's going to be susceptible to your infection, as well as showing similar infection to what's in humans. Rabbits and goats actually I've got here as well, and horses. They're quite often used for antibodies. So it's because they're larger animals, so you get more blood. For example, mice, you get like 1 ml of blood, which isn't enough if you wanted to generate a lot of antibody, you need larger animals. Whereas if you made an antibody in rabbits, you get a lot. And obviously even more in goats and horses. So it depends on what experiment you're doing and what disease that you might be looking at as to which animal you use. Ease and adaptability of experimental manipulation. So if you're working in PC3, for example, you don't want to be working with horses because they're big. You have to house them outside, and obviously you have the environment. It could be a risk to the environment. But I will talk to you about hazards of working and difficulties of working with PC3 in a minute. And of course there's the ecological consequences if an animal escapes, and ethical implications. And these are all considered and all have to be laid out in your ethics application in fine detail. So I'm going to talk about Burke. Burke can cause disease in absolutely any animal. I've got pictures of a gorilla here. In Singapore Zoo, they can't have gorillas. They tried twice. Both cases, the gorillas came down with melioidosis. So, and again, in Southeast Asia, the bug is in the soil. The second time they housed the gorillas, they housed them indoors, but they still managed to come down with melioidosis, they thought, from an air vent or something. I've got pictures of dolphins here in Hong Kong, Marina Park. The dolphins started dying of melioidosis, and so they trialled a heat-killed vaccine to try and protect them. They had some success. So it can affect fish as well. But it affects kangaroos, goats obviously, snakes. There is in Darwin a collaborator, Bart Curry. He's one of the pioneers, I would say, of melioidosis in terms of diagnostics and in terms of treatment. So in the Northern Territory, they get something like 100 cases per year. And for them, it's quite a risk because it is in the soil. So quite a lot of there's this street in Darwin where they joke about it, but basically, if you garden in that street, that you're likely to come down with melioidosis. So a lot of gardeners have come down with melioidosis. When I was in Darwin visiting Bart, the taxi driver was telling me about melioidosis because he just got the information out there, warning people, especially when there's floods. The bug comes into the water, into the surface. We always see a spike in cases. And this is also the case in Queensland as well. But there's this park, this animal park up there, and that park, they had like ferrets and reptiles and crocodiles, and all the animals at some stage came down with melioidosis as well. So it's really, it's such a, in terms of host, it's really versatile, affects humans, it can affect plants as well. So just a little bit about the disease. As I said, it causes melioidosis. It's the third most common cause of death in Thailand after HIV and tuberculosis. It causes about 40% mortality in Thailand. Here in Australia, it's about 14%. The thing with Burkholderia pseudomallei is that it's really highly antibiotic resistant. It has these efflux pumps, and it just pumps all the chemicals outside. So treatment is really hard. So treatment is 2 weeks intravenous therapy with like meropenem or ceftazidime, and then another at least 6 months, if not longer, oral eradication therapy with trimethoprim, sulfamethoxazole. It's really quite hard, and it causes acute, chronic, and latent infection, which I'll talk about in a minute. But here I've got a picture of rice paddy field workers. It's an occupational hazard for them because they have cuts and abrasions in their skin, and that's how they acquire the infection whilst working in these paddy fields. And so a lot of them, there's a lot of farmers that go presenting with the infection in not just Thailand, Vietnam, all around Southeast Asia. And so infection, well, there are the roots is most common by cutaneous, so the cuts and abrasions in the skin, but it can also be acquired by inhalation and ingestion as well. So infections can be asymptomatic, acute septicemia, you can get a chronic longer-term infection, and then you can get a latent infection. So one of the reasons why the military are interested in Burkholderia pseudomallei is because in the Vietnam War, quite a lot of soldiers came down with melioidosis, and this is because they hypothesize is because of the helicopters landing, aerosolizing the dust, and soldiers getting it. Now, there was one Vietnam soldier, American soldier, who got melioidosis, recovered, went back to America. 62 years later, it reoccurred. So it can hide away effectively. We don't know where it hides away. Some diseases like brucellosis, they know that the bacteria hides away in the tailbone of the mice, but we haven't discovered where Burke does that. So it can cause this latent infection. And risk factors include diabetes and renal disease. And this is all just showing you cutaneous melioidosis in the skin. So what are the animal models of Burkholderia pseudomallei? Well, these are the 4 that have been used: mice, diabetic rats, and there is an instance of diabetic mice as well. And this is to look at the susceptibility, the fact that if you're immunocompromised, you're more susceptible to melioidosis. Hamsters have been used because they're very susceptible, and also marmosets more recently. My colleagues at DSTL have been having a look at the marmoset model of melioidosis. Now the rule is, when it comes to screening drugs, the first lot of animals is rodents, usually mice, unless there isn't mice is not the appropriate model. But then you need to do a second model that's higher, or even higher order, so something like dogs. But most commonly in the past, people have been using monkeys. In America, they use like the African greens, but those experiments are really expensive to do. But we've used the mouse infection model. There are things that you need to take into consideration, like the model that's most appropriate for the work that you're going to do, health and safety, and then obviously the costs. The costs of housing marmosets and breeding marmosets is really quite high. Mice is a little bit cheaper. Health and safety wise, this Burkholderia animal model has to be done in PC3. So you have to think about personnel safety. When you're going into an animal model, you use injections, you're cutting up the animal, you know, so you need to, so there's sharps around, you need to be really careful and have really good, strong safety practices. So the mouse infection model of Burkholderia pseudomallii, there is two. There is two different species of mice. There's the Balb C and there's the C57 black 6s. Now, interestingly, the Balb Cs are highly susceptible to infection. So the Balb C is a really good model for acute melioidosis. The C57 black 6s are highly resistant. So that's a really good model for chronic melioidosis. So that's why the interspecies variability is really quite interesting as well. But it allows you to tease the different infection models. The other thing about Balb Cs is that they're really nice. They're very mild, nice temperament. Whereas the C57 black 6s are really feisty, and they have these temper tantrums, and they try and jump out the cage. And so, you know, so you need to take these things into consideration, especially when you're working in PC3. So also when you work in PC3, and this is the Biomedical Research Facility in Shenton Park, UWA's PC3 suite. There's two rooms. This one here is like a microbiology room where we do all the microbiology and processing. Here is the animal. But this, I've just taken this picture of the PPE that we have to wear. So we have to have these masks with these visors so that we don't get anything sprayed on our faces. We obviously have to wear hairnets, a lab coat, but then you have to wear a second lab coat over this. And then you have to be double gloved. You wear scrubs, so you don't wear your own clothes. And then you have to wear these overlays, shoe overlays, because you're working with animals. So it gets incredibly hot. So you really have to think quite carefully when you're designing these animal infection models. So this is the laboratory itself. So here we have this special cage, because obviously, even though there is very little evidence that Burkholderia pseudomallii is an inhalational risk, these animals are caged in closed cages, and so you have to, and they have their own air supply into each individual cage. So you have to have this special rack where each cage will go into, and each cage will be get its own air supply. And so everything is enclosed. You take it out of the rack, and then you put the cage in the cabinet, and then you do all your manipulations in the cabinet. So you have to be very careful of how you do the experiments. So obviously we need the animal ethics. Like I said, my latest one was 150 pages, but we managed to get it through. Welfare of animals are at the forefront. Monitoring, you have to go in and have a look at the animals at least twice a day, if not more, especially if they're not well. The numbers of animals, making the most of the experiments, making sure you get as much information as you can out of the experiments. And then the 3 R's, the replacement, refinement, and reduction. And then there's safety considerations. At PC3, 2 people have to be in there all the time. The supervisor, me, I'm on standby all the time. Injections and aerosols are the risks. So safety also has to come into this. So establishing the model at UWA. So when I came here and thought about developing the mouse model, it was mainly to screen these new inhibitors that we've got. But the first thing that I needed to do was to show that we could replicate the models. So what we did was we used C57 black 6s and bulb C mice species, 9 of each, and then we did we challenged IP, so that's intraperitoneal into their stomach, and we challenged intranasally. So we've got 2 different routes of challenge. So when we come in to screen drugs, we can use the intranasal and say, look, these drugs are effective against an inhalational challenge, inhalational melioidosis, or these drugs are effective against more like cuts and abrasions in the skin, IP. So we did 2 different routes, and we left the animals for 5 weeks. And this is what we saw. So this is a survival curve, and you can see that by the interperitoneal, we see that they succumb rapidly to infection, but we still had some animals left at the end of the experiment. With intranasal, we saw this, and we were quite surprised, because which route do you think would be more severe in terms of infection? Intranasal or IP? Yeah, intranasal, because you get pneumonic melioidosis. And so we were really surprised to see this, because we would have thought it would have been the other way around. But actually it turned out it's because of how we were doing the intranasal challenges, because at the time we opted to not knock the animals out, like give them a bit of anesthetic, because that was another additional step that would have been quite tricky with ethics, because they don't want you to do things like that, and so we didn't, and so we were just giving intranasal straight onto their nose and hoping they would breathe it in, and which they did, but clearly it's not as effective. So what we started doing after this, so remember the refinement of experiments, so this is why you do these first initial development studies, to refine your experiments, to make them better. So after this, what we do is we anesthetize them just for a minute or 2, and then put the drop on the nose, and they just breathe it in, and we've had beautiful results since. But that was one surprising result. And then with the animals that were left at the end of the experiments, we had a look to see if they had bacteria in their lung, liver, and spleens, and you can see that they do. Apart from this one, this cheeky mice managed to subvert it. But that's one of the features about animal models, is that it's with the Burke one especially, is that some animals, for some reason, manage to clear the infection, even though they're genetically meant to be exactly the same. But we do see that in the human population as well. Some people don't get melioidosis, whereas other people do. So with the bulb C57 black 6s, if you remember, these animals are less susceptible to melioidosis. And so what we saw was that all animals survived. They're all perfectly happy. They're all jumping around. And so there was no deaths at all in these groups. So what we did instead is we scored them. So when you come in to look at animals, you score them according to how well they're looking. So basically, is their fur ruffled? If their fur is ruffled, are they grooming themselves? That's one score. Because if their fur is ruffled, it means they're not grooming themselves, which means that they're not feeling happy. Are they haunched? So when they're feeling sick, they kind of like look a bit haunched. Are their eyes bright? We all get a bit sad when we're not feeling well. Our eyes aren't as bright either. So it's the same with animals. So we did, so this is just the scoring. And you can see that most animals scored 0, basically. Except that this animal here showed some signs, but then was fine again. But towards the end of the experiment, you can see we're seeing increasing signs of symptoms. But but at the end of the experiment, when we had a look at the organs, you can see that for IP challenge, they all had bacteremia. Clearly, even though some of these are quite high, if you look at it, about 10,000, despite having that much bacteria, these animals looked quite healthy. And then with the intranasal, we saw these 2 manage to clear infection, but the rest did have some bacteria. So once we've established that we can actually do, the other reason for doing that experiment was, one, to verify that we could do the model, but second was to help us understand how the procedure, how we were going to do certain things, how many people we need, how we were going to, little things like how do we hold the mice and inject the mice without injecting one of us. You know, it's all about technique and procedures and safety. So the next thing we wanted to do was look at the virulence of K96, strain K96, which is what we're going to use to challenge for our drug discovery study. So we want to know, from this experiment, what we wanted to know was what dose should we use to get an effective challenge. So we used BALB-Cs, N equals 6 in each group, and we did IP, intranasal challenge, but with differing levels of CFUs of K96. So we went from 3 times 10 to the 5 to 3 times 10 to the 1 for IP. We went a bit higher. For the intranasal, we went 3 times 10 to the 4 to 30. And this is what we saw. So for the IP challenge, you can see we saw a dose-dependent effect. So the ones that were given 10 to the 5 succumbed very quickly to infection. So this was clearly too high a dose to use for future experiments. The ones that were given lower, we see that they succumb but much later on. So whereas the one that was given 1,000, we see they all succumb to infection, but you can see that that we see a nicer curve and they don't die off quickly. So 10,000 is probably the challenge dose that you want to go with, where you give them enough bacteria so that they're sick, but not enough that they just cripple and die. Because the reason why this is important is because we're trying an antivirulence drug. So if we give too much bacteria with this antivirulence drug, we might not see an effect. So that's why the dosing is important. Well, the challenge dose is important. Same with intranasal, but again with the intranasal, we see that they, at 10 to the 4, they succumb quite quickly. But you get these lingerers, always get these lingerers that seem to be like fighting the disease, and so we get the odd animals that survive. With intranasal, we decided to go with 100 because again we see this nice effect where they're infected, but they're not dying super quickly. So when we were thinking about how we're going to screen the drugs that we have, there are 2 models that you could do. One is, do you dose with the drug and then do you just leave the infection to run and see if the mice survive at the end of the experiment? Or the other way to do it is to challenge, but then dose with drug and look to see if you see a reduction in bacteria in the lungs. So because our therapeutic is really in its early stages, and because it's an antivirulence compound, we decided to look at bacterial numbers. So to validate the model, we decided to do 0 to 7 days, and we wanted to see what the bacterial burdens look like in the lung, liver, spleen, kidneys, and blood. What we found when we did the actual experiment, that removing blood from animals was quite tricky, and also the data was wasn't great, because it all depended on how much blood you can take out, and quite often IP, when it goes in IP, you don't see much bacteremia in the blood, whereas in the lungs, livers, and spleens, and to a degree in the kidneys, you see quite a lot of Burke in there. So here, what we did was we had groups that we took each of these days, and the aim was to check for reduction clearance of bacteria. So this was the experiment that we did. So the animals, when they come in, they have to acclimatize for a week before you can do anything, so that they get comfortable in their new environment. And then we challenge on day 0, and then we took each group and each of these days, and had a look at bacteria in the various lungs. And so this was performed by the IP route, And you can see nice increase in numbers of bacteria by day 7 in the spleen. In the liver, you can see that there is a variable amount of bacteria, but there is bacteria in more. In the lung, kind of similar to the spleen, you see bacteremia, but it stays fairly static for the 7 days. And in the kidney, a bit like the liver, a little bit varying levels. And you get these odd mice where, you know, there's hardly any. It's like the infection's not doing much to them. But you can see that there is definitely bacteria in these organs during each of those days. So this is by the IP route. And we saw a similar sort of thing by the intranasal route. And so what we did see with intranasal route was, look at what happens in the lungs. You see nice level of bacteria, but you see that increase that you do with the spleens. So the two organs that show the most really is the spleen and the lung. So then in terms of developing the model further, so remember I was saying about our drugs were antivirulants, so we want to have a look to see if the drugs on their own have an effect, but also wanted to have a look if the drugs have an effect in combination with antibiotics. So in order to develop that model, we chose the antibiotic doxycycline. Doxycycline isn't used as the standard treatment for melioidosis, but the military really like doxycycline because it's a one pill saves everything. So we have done quite a lot of screening of doxycycline against Burke. And so in order to develop this model, we thought we'd do a suboptimal drug efficacy, because you don't want it to be optimal because it'll be the doxycycline that treats. What you want to do is show that the doxycycline is highly effective with the drug drug that you're testing. And so what we did here was, again, we had the acclimatization period, we challenged the animals, and then every day we treated with doxycycline. And at each each day we took a group and we had a look at bacterial numbers. And so this is with the IP challenge route. You can see that in the red are the untreated animals. We see increase in bacterial burdens in the spleen, but with doxycycline treatment we see a lot less. Same in the liver, similar in the lung, maybe not quite as clear, but remember we are injecting these animals by IP, and then in the kidney as well. This one we did exactly the same, but by the intranasal route, so that we've got that option of treatment by IP or treatment by inhalational. And you can see exactly the same. So in the spleen, doxycycline treatment, we see less bacteria. Here you see a beautiful clear differentiation in the lung, in the liver, and in the kidney as well. So I was talking about refinement. So with the doxycycline, you actually deliver it orally. And so this is what oral gavage is. And initially we were doing this method because this is not a needle. It is a needle, but there is a metal bulb at the end of it, so that literally it just slides into the mice and goes to the stomach, and you deliver the whole entire drug in their stomach. And we found this was fine for the animals that weren't sick, but for the animals that were sick, it was giving them a lot of bother. So we refined it by using mouth pipetting. So what we did was we mixed in a bit of sugar with their doxycycline, and they just and so this is what I mean about refinement. Like when we were doing it, we realized that the animals were in distress. This isn't a good method, so we refined it to doing the mouth pipetting with sugar-coated doxycycline. And not only was it less distressing to the sick mice, but it was a lot quicker as well. They really did like the sugary solution. And so when we refined it, what we showed by the intranasal route was that we see exactly the same pattern. So even though we made that refinement, we had to put in a modification to our ethics application so that we could prove that the mouth pipetting was as good as the oral gavage. So what's next? Well, we're going to screen novel compounds, which is really very exciting. That's what we're going to do in the next few months. But I hope to highlight how the use of models whose physiology and genetics are closer to humans is really important. You have to think very carefully about which model you're using. The FDA did have a 2-model policy that I mentioned. However, I came across this that says that the FDA no longer needs to require animal tests before human drug trials. So that's quite a big step. However, I think this is not going to come about tomorrow, because you still have to prove that the drugs are going to be safe. So, so obviously this will have made the animal advocates very happy. I will say that with the animal advocates, some of the paraphernalia they use, those are of images of animals in the 1950s and 60s where there was no regulation of animal studies. Nowadays, the animals are actually better looked after than some children, to put it quite frankly. And if you're housing 2 rabbits, the size of the room that you have to house them in is actually really, really big, bigger than some rooms in London flats. So you know, so the regulations are there to help make sure that the animals in the short lifespan are happy. So researchers say not all drug screening are the same. Different diseases require different models for validation. However, it has opened up a path for alternative models. So these are the organoid models, the 3D tissue culture models, all those sort of things are becoming more popular. So it'll probably take a while with lots of testing and validating of the different systems. So that's it from me. Thank you very much for your attention. Got any questions, or do you want me to? Yes, fire away.